Enteric viruses infect our gastrointestinal tract. The most famous one are Norovirus and Rotaviruses or Astroviruses. So while we have a lot of vaccine available for rotaviruses, these vaccines are still very inefficient in developing countries, and we still try to understand why these vaccines are not efficient and how can we actually make this vaccine better.
So the gut as this massive interface between external and internal environment is really important for balancing protection against enteric pathogens, but then also tolerance of food and commensal microbiota. And it does this through this really unique oxygen environment known as hypoxia, where you have low oxygen.
For example, rotavirus, which is a major cause of diarrhea in infants, infect the very top of the villi, which is where there is no oxygen. So if we understand how oxygen regulates the response of these cells to rotavirus, we will be able to create a specific treatment to stop rotavirus infection.
We use a lot of different techniques to sort of take that hypoxic environment and bring it to cell culture. So we have a glove box, which is able to maintain our cells at 1% oxygen. And we also use a microscope, which allows us to do live cell studies of cells in low oxygen. So we can really see how that natural environment is being replicated in a cell culture model.
So one of the main findings we have achieved recently is demonstrating that hypoxia is able to control the immune response generated by intestinal epithelium cells, so making enteric viruses more infectious. Understanding how hypoxia regulates the immune system to favor virus infection give us a really fundamental cue in the mechanisms used by a cell to control pathogen. So by exploiting these mechanisms, we can develop specific drugs to activate this pathway and now target very specific viruses in the gut in order to control an infection.